It is my pleasure to introduce our next speaker, Kwabena Boahen. Kwabena is an associate professor of bioengineering at Stanford University. He is currently using silicon integrated circuits to emulate neural computation. Please join me in welcoming Professor Boahen.
Thank you. Can you hear me? Is that okay? No? I think we need to turn it up a little bit. Okay. Hello? Hello? Yeah, it's on. Okay. This is better? Or maybe I need to move it in. Yeah. Hello? Hello? Is that better? Yeah, okay. Thanks. Okay, so actually I started advancing stuff. But this is good. Okay. So the motivation, basically in terms of what we've been doing, we've been inspired by the efficiency of the brain at doing computation. And I'm going to go over a few numbers here, just a little bit of back of the envelope calculation to compare how efficient the brain is at doing computation and a computer. And so here, a person at rest, just sitting in this lecture hall, not doing any real work, you consume about 100 watts, which corresponds to a 1,400 calorie a day diet. And 20% of that energy based on oxygen supply is going to your brain. So the brain is using about 20 watts. And that then number, 20 joules per second, allows us to calculate how much energy per operation the brain is using. So here you see there's something like 10 to the 12 over a trillion neurons in the brain. Each of them make about 1,000 connections. And each of those connections is activated about 10 times a second. So that gives you something like 10 to the 16 synaptic operations per second. Okay, and so these synaptic events, when you fire these synapses, are the most primitive or the fundamental operation that's going on in the brain. And so if you take those two numbers, the 20 watts, I'll round this down to 10 joules, and 10 to the 16 operations with 10 joules, you get 10 to the minus 15 joules per operation. And if you compare that with what Blue Gene, the fastest computer in the world right now, and your chair was one of the lead architects on this project before he came here from IBM, Mark Snare. And the original design, this slide was the original design of the machine. The actual machine they have now is a little bit different from this, but the idea was to basically build a machine with 1 million processors. And so there's 1 million processors in there, and each of those processors would give you a gigaflop. And a gigaflop times 1 million gives you 10 to the 15 operations per second. So the goal was to get a petaflop machine. And that right now they are at something like about 100 teraflops. And the computer was designed to consume 1 megawatt of power, which is actually about 2 orders of magnitude more efficient than what you have in your Pentium or on your desk, because your desktop computer is a gigaflop and it's about 100 watts. A petaflop at 1 megawatt is about 1 watt per gigaflop rather than 100 watts. And so that works out to 10 to the minus 9 joules per operation. And so here you have a 6 order of magnitude difference between the fundamental operation that a brain is doing, actifying a synapse, and the fundamental operation that a computer is doing, executing an instruction. To a certain extent, I'm comparing apples and oranges here, because these are maybe 64-bit floating point operations, and these are just activating a synapse. But it really doesn't matter, because if you wanted to program your computer to do this, you can't use any less energy than this per operation. You'd have to translate whatever you're trying to do into these 64-bit operations, whether you like it or not. Okay, Mark Snare made that decision for you. You don't have any choice, right? Whereas the brain has made a different kind of decision. And somehow it seems like it's much more effective at using these operations to do the computation than we are at using these big machines to do computation. At least the kinds of computations that I'm interested in, like recognizing a face and the kinds of things that biological organisms excel at. And so this comparison was done actually in the mid-'80s by Carver Mead. And based on this, he started a research program, or he basically redirected his research to trying to develop what he called neuromorphic electronic systems. Okay, and so the idea was to model neural circuitry in silicon. In other words, use the same transistors that we are using to build these computers, but somehow organize them differently based on how the circuits are organized in the brain. And one of the most successful projects that they did was this silicon retina, where you see here the output of the silicon retina looking at a cat on the Scientific American from 1991. I'll get back to that later, but this is just to give you the idea of the concept was to actually translate these neural circuits directly into silicon circuits, something that we call morphing. And the reason why Carver felt that we could close this 6 order of magnitude efficiency gap in energy dissipation between computers and the brain was that when you look down at the device level and you compare the devices that neurons use and the devices that we use to build computers, the energy is comparable. The gap is not as big. Okay? And so here what you do is you take the elements that neurons use, like these ion channels. They are little protein pores that sit inside the membrane of a neuron, and they allow ions to flow. And this then generates electrical activity, then which leads to all these signals going back and forth between the neurons and so on. And I've just put some numbers here that I've used to calculate how fast an ion channel can switch and how much energy it uses to switch. And so it takes about something on the order of 10 nanoseconds for an ion to traverse that go from inside to outside the cell. And the energy it takes to activate a contact between two neurons, about 1 micron squared synapse, is on the order of 10 to the minus 20 joules. Okay. And if you compare that to the transistors that we are building in 130 nanometer technology, this is about a factor of 2 bigger than what we are doing now. And you use those numbers here, it turns out that, transistors electrons move a lot faster because electrons have about 1 million times the mobility of ions. And the amount of energy that it takes to switch one of these transistors on and off is about 10 to the minus 18 joules. Okay? And so that number there is only 2 orders of magnitude bigger than the other number here, 10 to the minus 20 joules to activate a synapse. Okay, it's just about a factor of 100, not a factor of 1 million like we saw at the system level. And also, in fact, these devices, if you compare the speed, are much faster. Okay, about 10 to the 4 times faster for these electrons to cross than the ions to cross. And so you can actually scale down the power supply because the switching energy goes like the voltage squared by a factor of 10, and you can match the switching energy, and you can still be a factor of 1,000 faster in these devices because the transit time only goes like the inverse of the voltage. Okay. And so these numbers convinced Carver that actually we use these transistors. The transistors themselves are pretty good devices, and maybe the way we are using them is not so efficient. Okay. And so this made him argue that he could close the gap. And so now, where does all that energy go? We start out with these transistors, and they are pretty similar switching energy to synapses. But by the time we do some useful computation, we are using about 1 million times more power, okay, at the system level. And so here is just graphing a little bit. So we start out here. This is my energy scale. So to fire a synapse, like I said, it's about 10 to the minus 15. To actually, at the system level, that is, when you take into account all the wiring that's connecting these neurons together, and all that, switching all that on and off, it's about 10 to the minus 15. If you just look at the contact itself, 1 square micron, it's 10 to the minus 18. Sorry, that was on the previous slide. But, this is everything is an order of magnitude. And here, when we switch a transistor, we are saying we are down here at about 10 to the minus 18. 10 to the minus, an individual transistor about 10 to the minus 18 is down here. And as we basically do more operations. So in an ASIC, just like driving a bus, a 32-bit bus, across the chip, you consume about 10 to the minus 12 joules. In an FPGA, doing an ALU operation type thing, It's another order of magnitude. By the time you get to a PC where you're going off chip and you're switching between chips, you are on the order of 10 to the minus 9 joules per operation, which is the number that we had, versus 10 to the minus 15 for an operation in the brain. Okay. And so basically the answer is simple. The reason is, again, I'm going to switch this mic over to this side because I tend to turn in that direction. And maybe go. And so the reason why, oh, okay, I need to go back. And so the reason for this increase in energy, the reason why we are squandering all this energy is simple, is that we are using more transistors per operation. We're switching more and more transistors to do a single operation. And as we go up this chain, that number is increasing. And that's why we are burning all this power. And so this is the motor. And so my take on this, I'm saying I am buying the whole argument that Carver is making, Carver Mead made, about the actual devices being pretty efficient and the way we are squandering the energy and trying to now wire up those transistors in a more efficient way that is basically customized to do the job that we're trying to do is something that he didn't come up with an answer for. Okay? And this is where I come in with this concept of metamorphing. And I think that the fundamental reason why we're using a lot more transistors to do these operations is that that's how we manage complexity. We use abstraction to manage complexity. Okay, so when you're writing your program in C and you're doing some operation and it's being compiled down to assembly language, it's going down to the transistors, by the time, at the end of the day, when those electrons do the work, as Yale said, there's a lot of electrons flowing around to do the work. Okay, but then if we had to go and connect one of those, tell each one of those electrons what to do the work individually as we're programming, well, that's going to be too complex a task, right? So the way we manage that complexity is to abstract. Okay, and so this abstraction, in a sense, is failing to manage complexity, or in other words, we are paying this price of a huge inefficiency in power dissipation by going that route. And so that's sort of this. I like this quote here from Scott Watson from this website, the Tech Report. When Gordon Moore's goose lays a golden egg, and the number of transistors possible on a chip doubles, as it's supposed to do every 18 months, taking advantage of the windfall has proven increasingly difficult. Okay, so we're getting more and more transistors on the chip, but we don't know what to do with them. Okay, the old way of doing things, where we basically try to get the chip to run twice as fast every generation, Intel is telling you that that's not going to happen. In 2004, they canceled the 4 gigahertz Pentium. And now they're trying to convince you that what you need is a dual, a quad, or whatever, right? And like Yale was arguing, well, is that really going to do it for you? Okay? But the point is that it's not going to get any faster. And so is there some way to really take advantage of this windfall that we have twice as many transistors? We can't run them twice as fast, but can we actually use them to do twice as much computation? Okay. Now, nature has come up with alternative strategies for managing complexity. In fact, brains program themselves at the levels of individual neuronal connections. So they are doing exactly what we are talking about. They are putting the wiring in there, customizing the quote-unquote chip to do the computation. And these are individual connections that are being programmed rather than one instruction for the entire machine, which has to be supplied by a programmer, not by the brain itself. And so I'm going to try and show in the rest of the talk some progress that we've made in actually trying to use these transistors to do the job based on customizing the circuitry using the concepts or the principles that biology uses to manage complexity. And so this will tie in quite a bit with the previous talk that you had about connecting the biology with the engineering. And so just to give you some history, those of you who don't know, we've been doing this business for 40 years. We've been doubling transistors on a chip, every year and a half to every 2 years. And this has been going on for 40 years. And the levels of abstraction have kept increasing and increasing to handle that, to manage that complexity. So we started back in the 50s with these rubellite sheets. Okay, these are like mica sheets, and you actually take a knife and you cut out the patterns, and you expose them, and that's how you make your chips. And so you have these things laying on the floor, a huge amount of them. And then we came into transistor level design. So here you were actually medium scale integration in the 70s, and a lot of these companies are no longer around that made these tools. In the 80s, you had gate level schematics. So you had these, you were designing at the gate level, hooking gates together. In the 90s, you started going to VHDL, Verilog, high level synthesis, where there's whole registers, data paths. There's all these sort of more complicated blocks. And now in the 2000s, we are talking about IP blocks, right? You can get a whole processor chunk from somebody, and you can get all these bits and pieces, and you are really designing at the system level. This is what we've done so that we can manage the complexity, so that we don't have to figure out what every single transistor should do, right? You just lay them down in bigger chunks and bigger blocks, and you're not customizing at the transistor level. And yet, so despite all this, effort in the CAD industry, there's still an increasing gap between the amount of transistors that we can actually design and the rate at which these numbers of transistors are growing. So this is sort of Moore's law. The logic transistors per chip is plotted on this axis. It's been going at a rate about 58% per year. And then this is the number of transistors per staff per month. So how many transistors basically these chip designers can generate every month. And that has been going at a 21% rate. And so this gap just keeps increasing despite trying to work at higher and higher levels of abstraction. So abstraction is not really keeping up with it. And like I showed you in the first slide, it's really costing us a lot in energy efficiency. And if we keep doing this, the projected cost of a $130 million transistor chip is going to be, about that much. And so how does the brain do it? How does the brain deal with complexity? How does it manage complexity? I told you that we have 10 to the 12 neurons in the brain, which is way more, that's a trillion, way more than the number of transistors we have on the chip. And if you look at the number of neurons that a fly has, it's about 100,000. In our brain, we have 10 to the 12. Now, if you look at how many genes a fly has in its DNA, there are about 13,000 genes. How many do humans have? Anybody know? What? 25,000. That's exactly. I thought I hit the button by mistake. Yeah, 25,000. Okay, and there were bets on this number, right? You all know about the Human Genome Project, and there were bets on this number. People were saying that we got about 100,000, and so on and so forth, and nobody expected it to come in a factor of 2 from the fly. Because you got 10 to the 12, these are the numbers, right? We've got 7 orders of magnitude more neurons just by doubling the number of genes in the DNA. Okay? And so we basically, biology has managed to increase the complexity 7 orders of magnitude, and it's done it just by doubling the recipe for building the machine, the self-replicating machine. Okay, and we still need some DNA for the stomach and for all this other stuff, right? Okay, we're not using all this for the brain. Okay. And so I think that this is something that engineers can benefit from, right? The idea is that there's all this developmental machinery that we got a little tutorial on from the previous talk about RepRap, inspiration for RepRap, that really can generate a lot of complexity, and it really hides it so that you can start with a very simple description and you can end up with a much more complex machine. And so, just to push the analogy further, this is what's happening with the CPU design. You started out with the 4004. It had 23,000 transistors in 1971. It was one engineer designed that with the rubylith and cutting up the Mylar and all that stuff. And now we've got the Pentium 4. This is from 2004. With Intel keeps this a secret, right? When they announce, they try to intimidate everybody. They say it cost us $4 billion to design this chip, and you didn't even try it, and so on and so forth. So we don't really know. But my estimate is maybe 800 engineers went into designing that. And you can see here, all this is memory, right? So part of what we've done to manage the complexity is not to even try to do anything with those transistors, just add more memory on the chip. And even though we've only gone, 4 orders of magnitude in number of transistors, we've gone two or three orders of magnitude in numbers of engineers. Okay, if you think of engineers as genes, you know, the biology is doing a lot better. Just doubling the engineers, have got 7 orders of magnitude more complex devices. If we go at this rate to get 10 to the 12 transistors on a chip, it will be a small city, okay, of engineers to do that. Okay, and so we've got the devices, and we've got all this great technology. We don't really know what to do with it. Okay, or we can't afford to do anything with it. And so that right now, you know, following on Carver Mead's inspiration about trying to actually use the biological paradigm or the neural paradigm to do computation, that's sort of grown. It's still quite a small community, but this is just a list of different people who are now trying to use transistors in a different way to directly model different kinds of neural computation. And there's different universities listed here, and people are looking at motion sensing in the silicon retina, all the way to learning chips, and so on and so forth, for seeing depth and so on. And so I'll tell you a little bit about, you know, sort of the classic, a little bit more about the silicon retina that I mentioned briefly earlier on. And then I'll tell you what we've been trying to do more recently is to actually, instead of trying to model these circuits directly, translate neural circuits into silicon circuits, we want to translate these developmental algorithms into silicon chips so that now we can use the same approach to manage complexity, okay? And we've done some initial steps in that direction. So you see that at the end. But just to show you where we're coming from, I want to tell you about a little bit more detail about how you take the retina and you translate that into a chip. That's designing at the circuit level. And then I'm going to talk about designing at the developmental level, okay, which is more of a computer sciencey type of thing, right? And so just a little bit about the retina. The retina is basically the film that lines the inside of your eyeball. It's got 5 layers that you can see here. The ganglion cells are the output. The light comes in here. These are the photodetectors. It travels through these layers, and it goes down the optic nerve as electrical pulses or impulses. Okay. And these 5 layers then give rise, the circuitry in these embedded in these 5 layers then gives rise to these selective responses of the ganglion cells. So you can think of it as a camera. Okay, it's not just sending out the raw intensity. It's generating a whole bunch of different pictures that reflect what's dark or what's bright, what's moving, and so on and so forth. And there's these guys here which have small receptor fields, which means that they have high resolution. They just some intensity over very small areas. And there's these guys here which have large receptor fields. And these guys respond in a transient fashion. So, you know, either when this central region gets dark, which is shown here, when it gets bright, they don't respond. And when it gets dark, you get a burst of spikes and then silence. Okay, these are the impulses that travel down the optic nerve. And you've got the counterpart, the complementary cell here, which when the central part here brightens, you get a burst of spikes and then silence, and so on. And then these guys are sort of complementary. They are small. They want small spots, not large ones, but they respond in a sustained fashion. So they keep firing for the entire duration of that spot, either darker or brighter. And these 4 particular types of ganglion cells constitute 90% of your optic nerve. So you've got about 1 million fibers in your optic nerve. 900,000 of those are these 4 types. The remaining 100,000 are another about 8 different types of cells, which are doing things like, you know, when your eyeball rotates, you have to sort of, when the scene rotates or you move your head, you have to move your eyeball to compensate. These things are being computed. That motion is being computed directly in the retina, and it goes straight to your oculomotor system, reflex. And so we don't care about those guys so much. We care about these guys who are going to go to cortex, and that's how you're going to recognize objects and things like that. And so the basic idea is to take the information that the neurophysiologists, neuroanatomists have figured out about these interactions happening in these 5 cell layers of the retina and translate those circuits or that synaptic organization into an electrical circuit or a transistor circuit. And the basic way to do that is to come up with primitives. So you have synapses between cells. You can come up with a way of implementing a synapse with a transistor. And then you can wire up your circuit. And so here is one way of doing that, where you have a cell here which comes and it inhibits another cell, and that connection is modulated by a 3rd cell. There's a lot of this stuff going on biologically, right? Biological systems are always adapting. They're always internally controlling and modifying their behavior based on what's happening, okay? It's all autonomous. And so the modulation is important. And you can do that with a transistor. You can set the voltage on this gate of the transistor to reflect the voltage of this cell. And then you can take the current that flows in the transistor and you can connect it. You can take current off of a node whose voltage represents the activity of that target cell. And so this will discharge the voltage, and that cell's voltage will reduce, and that corresponds to inhibition. And you can permute the connections. You can do it like this. You get an excitatory synapse. And you can do it like that, and you get a gap junction or just a conductance that just goes both ways between the two cells. And furthermore, what we do is we operate these transistors down in the sub-threshold region, which is a region where you are taught that the transistor is off. You go below the threshold, the transistor is off. It's not actually off. If you plot on a log-log scale, you see that once you get below the threshold voltage, this is about a volt, this is an old process, there's an exponential decrease in the currents. People are now more aware of this because of the off currents, and the leakage currents are now 10% of the power dissipation in these chips. So people are worried about that. But there's an exponential decrease, and the current changes from picoamps to about microamps, where then it starts going square law, which is the above threshold behavior. In this regime, these picoamps and nanoamps are the same kinds of currents that these neurons are talking to each other with. So when a synapse activates, opens ion channels in the target cell, each ion channel passes about a picoamp. About 100 of them together it takes to fire the cell, passes about 1/10 of a nanoamp. And you can get the similar type current levels here by operating transistors in this regime. It's your standard CMOS technology. There's nothing different. And so using those primitives and using the circuitry that the biologists have figured out about the retina, we were able to actually model all the 5 layers of the retina and get these 4 different types of outputs that constitute 90% of the optic nerve. This was Carver's original retina. It only did the first 3 layers. And so this actually is really a complete model of the retina. And so what you're going to see here is the outputs of these 4 different types of ganglion cells in color, where the color code red and green corresponds to cells that like dark. They like the dark spot in the gray background, the red guys, and the green guys like the bright spot in the gray background. They like contrast. And these are the sustained guys. They keep firing so long as it's darker or it's brighter. And then you've also got the transient guys who like it to get dark, the blue guys, and like to get bright, the yellow. Actually, it's the other way. It's getting dark here, so the yellow guys are responding. He's moving his head to the right. And it's getting bright here because his dark hair is revealing the white background. And you get the blue guys. So this is the code here. You've got guys who like dark, bright, or dark contrast to respond in a sustained or transient fashion. And so what you're going to see come up here is a video where you're going to see these kinds of color-coded images down here, and you're going to see a reconstruction from the spike train of what was actually being shown to the chip. So you can see this type of retinal processing generates a very sparse output because you only respond to contrast and to motion. But you can reconstruct essentially the image. I'm not going to get into how you do that, but you have to take into account the kind of processing that's happening in the retina. And here it's about a 50 by 30 pixel image. It's not very high resolution. But Kareem is almost recognizable from that picture. And this chip consumes, it's got, like I said, it's about 50 by 30 resolution. The total number of outputs is less than 4,000. And it's about 60 milliwatts of power dissipation from that chip doing all this computation directly using the transistors. And so that's the approach taken there is an example of the neuromorphic approach, where you look at the circuitry that evolution has basically evolved to solve these kinds of problems. And you can basically then translate that into a silicon circuit once you come up with ways of implementing these biological primitives directly with transistors. And you can imagine some of this is happening now. These kinds of devices, because of their low power, a couple of milliwatts, you can actually implant a chip like that inside somebody's eye if they are blind to replace the retina. But we're sort of pushing forward from that, where we want to build more complex systems, not just stop at the retina. We want to be able to model the cortex. Okay? And that's where we get into the complexity issue that we are facing with our transistors, and that biology is also facing. We're trying to build a cortex with 10 to the 12 neurons in it. And so now we are trying to actually do this by modeling at the developmental level, taking the approach of trying to implement those rules that biology uses to generate those circuits rather than the circuits themselves. And so here is one of the best understood examples of development, okay, which is activity-dependent development. And this is a classic system that's studied by biologists, where they study in these tadpoles of frogs how the eye wires itself up to the tectum. Okay, the tectum in us corresponds to the superior colliculus. Is this brainstem nuclei that controls your eye movements. So you're constantly moving your eyes, and it picks the targets that you move them to. And you'd imagine that you want to have an orderly connection from the retina to the tectum so that when you see something over here, you can move your eye to that location. But actually what happens if you go in there and you look at this process as the neurons are growing in, this optic nerve connections are coming in to the tectum, you find out that if you stain a small region of the retina with dye, and you allow that dye to be taken up by the neurons, and you find out where it ends up in the tectum, a small region of the retina actually lights up almost the entire tectum. Okay, so these neurons grow in there, and they actually don't know where they are going, and they end up in random locations. But then over time, the wiring refines itself, just based on activity. It does it just in a spontaneous, just the spontaneous activity that's generated, and you get now a focal, the same guys who ended up all here. You've removed all these wrong connections, and you've narrowed it down to a tight spot, so that neighboring neurons from the retina end up next to each other in the tectum. And this process, I just show you this little movie up here. This is a neuron that in culture that has just been born. It starts putting out these processes. One of them, the longest one, becomes the output of that neuron called the axon. These are called the dendrites. They become the inputs. And that thing continues to grow, and that's how this whole process is driven. So that's the summary there. So you see that in the movie. At the tip of the little axon is a little hand-like process called a growth cone that is feeling its environment, and it's growing. This is the whole thing will take about 24 hours. It's sped up. Okay. And so that's how these guys grow in there, and somehow over time they refine, they try different connections, they retract, and so on and so forth, and they get the right mapping. And so we modeled this process in silicon. Okay. And the basic idea is that we want to basically be able to rewire these connections between two chips so that we can start up with some scrambled wiring and make it basically a regular mapping. I mean, this is sort of a toy problem, right? We could have just done that in the beginning. But I'll show you, once we're able to achieve this, the same rules can generate other kinds of patterns. Okay. But so now you have this problem as to how do you actually make some wires, rewire some connections. When we make chips, we don't have these growth cones, we have these metal wires. And they're not going anywhere, right? And so what we've done is we had to basically do some combination of virtual and real wiring. Okay, so this is what we call soft wires. So the idea is if we wanted to model this mapping from the retina to the tectum, we got some wiring pattern. We basically assign each of our neurons on our retina chip an address, so that when it spikes, we send that address off the chip. And the retina chip I showed you before actually does this. There's streams of addresses coming up, and each time I light up a colored dot on the image, it's because I saw that address. Okay? And because there's 4,000 outputs coming out, and we don't have 4,000 pins. Okay? So we have to multiplex. So we're doing that. And so now, but because we're doing this, now we have these addresses. We can actually then take those addresses, instead of sending them straight to this chip, we can take them through a RAM, where we use them to look up what's stored in the RAM, and then we present that address to the second chip. And then that address then will determine which synapse we activate on this chip. Okay? And so I'm just going to animate this right now. So if you light up a few cells there, we'll go to the lookup table, we'll light up those cells. And now we want to rewire the connections. And what the biologists have figured out is going on is that neurons release a diffusible substance called neurotrophin, which diffuses across the tissue. And these growth cones can sense the gradient of this chemical. Okay? So at their tips, they have little receptors for these chemicals, and they bind them, and they figure out how much concentration is coming in. And based on that, they may decide to move towards where the chemical, up the gradient, towards where the chemical is coming from. And we also then want to implement circuits on our chip that, first of all, will model this neurotrophin as just charge diffusing across the chip. Okay? And then we'll have circuits on our chip that can determine the gradient of that charge. And then we want to move up that gradient. Okay? And so then the way we do that is that then we send this pair of addresses to the RAM chip, and we swap those two guys. That would affect the movement on the chip, so that now when this neuron 1 fires, it goes to the synapse 2 rather than to synapse 3 it was going to before. And so it's a combination of hardware and software, but not really software. There's no computer. You don't really need this. You can just use an FPGA or CPLD to implement the logic to do this. There's no real software involved in here. Okay? It's just remapping the addresses. And this is very similar to packet switching, those of you communications guys out there. Okay? It's a virtual connectivity. And so I'll show you some results from this chip here in a color mapped like that. Okay? So the idea is that you start up with a set of scrambled connections, and you just present patches of activity that look like this. It's in 2D. And so I highlight the guys here. Initially, the connections are scrambled so that these guys here end up all over the place, like you can see here. And you can track how well the mapping is because neighboring locations have the same color here. So what you like to have is neighboring locations to have the same color here. This is the output map in the target layer, and this is the input map on the input side. So once it's scrambled here, like you can see here, and it should eventually end up like that, so it looks like the input side. And so the idea, the biologists talk about these neurons that fire together, wire together. Okay, so what's happening is that when you're stimulating these patches, which is randomly generated activity in the retina, even before the eyes open, generates these kinds of activity. And just by neurons that are firing together, wiring together, you can refine this mapping, okay, and translate temporal coincidence into spatial coincidence. And so what you see here is a little movie where you're going to see it starting from this state here, and you're going to see presentations here, and eventually you're going to see the refinement of the colors. And so this is real data from a chip. I'm just presenting the connectivity map with these colors just to visualize it. And this is also time-lapse, right? We've compressed everything. But there's maybe about 8,000 presentations that go into generating this movie. And so that was the initial condition, and this is the current state of the wiring, the mapping. And so you can see we almost get sort of the quadrant-type organization. And in particular, this patch here is more concentrated on this direction, in this over here. And we got a few stragglers out there that lost their way, and we just have to kill those guys. But that would happen. That's what will happen biologically, but we don't have that in the chip. Okay. So that's the idea of one class of self-organizing or developmental rules that biology uses, which is called this activity-dependent development. And the general rule is cells that fire together, wire together. And by doing that, you can actually get this topographic mapping. You can also, when you have different cell types, like the on and off cells in the retina, or the left and right eye inputs, you can get other features like orientation maps, cells that are sensitive to depth, and so on. So the same rule can give you these things based on different types of input. Okay, but we haven't really demonstrated that yet, but people have done that in software. So we think we'll get there. But we've looked at the other class of developmental rules, which are the non-activity dependent or activity independent rules. Okay, and that's the second piece of metamorphing that we've done. And so here I have to tell you a little bit about how the cortex is organized. And so here you have your two eyes. You have the optic nerve going through something called the thalamus. It's a relay station. And then from there, you have neurons that project to the cortex. And back here in the back of your head is the visual cortex, the primary visual cortex, where sensory information first enters the cortex. You got them on the left side and the right side, and it's crossed, so the right visual field maps to the left side and vice versa. But you've got regions here where you have overlap of the representation. So you've got cells here which are getting both inputs from both eyes. And those are binocular cells. And if you go in there and you unroll the sheet of cortex, flatten it out, and you go in there and you record from individual cells, you'll find out that they actually like bars of different orientations, which is color-coded in this map. Okay, so so you find a region where you have all these red cells like horizontal, and then you find these green cells like vertical, and so on. Okay, and as you move along the cortex, that changes smoothly. Okay, and you also then switch from one eye to the other. You find cells that are driven by the right eye, then you find cells that are driven by the left eye, and so on. Okay, the important thing about it is that this sheet, you are tiling visual space, right? Every point in visual space maps onto this 2D sheet. But at the same time, you're also tiling different properties, like if it's a vertical bar, then a particular subgroup of those cells that respond to that part of visual space will respond, and so on. And so these are what are called feature maps, and they were developed by Hubel and Wiesel in the 70s, and they got the Nobel Prize for that. And I just listed some of the properties here that these cells respond to. Now, Hubel and Wiesel didn't really figure out, although they looked at development, we still haven't quite figured out how you build these maps because they seem very complex and very elaborate. And the activity-dependent rules I showed you before, this is pretty tough. And you can do it in software, but you need a lot of precision, and you don't have that kind of precision biologically. And so Alan Turing had proposed way back in 1950 a way in which you could form complex patterns just spontaneously from noise. Okay, and actually he was interested in explaining, how the zebra gets its stripes and things like that, right? And so his idea was that you could just basically start out with this tissue and have cells make dye, right? So you start out with everything is white, and you have some cells just decide randomly whether they're going to make dye or not, and you start making black dye and darkening tissue. And then you have cells also make bleach, which will then go and sort of bleach this dye, right? And if you had the right conditions, like you had the dye and the bleach spread at different rates, right? You could set it up so that you get these patterns which look like stripes or like spots and things like that. Okay, where the spots exactly form, you don't control. That just depends on some cell getting a head start and making a little bit more dye than bleach, right? But the overall structure of the pattern that you have spots and they repeat and so on, you control by how the diffusion properties of the dye and the bleach. Okay, and then stripes too, you can control. And these kind of random seeding of these patterns, just randomly making some cells make a little bit more, a little bit less, you can get for free because we now know that cells, even though they have the same DNA and are clones of each other, when you tell them to make the protein that those genes are expressing, they make different amounts. The expression is stochastic. So these are all E. coli bacteria that are all identical, but yet make different amounts of this fluorescent protein. Okay, they are clones. And so you can see the pattern just by this noise, stochastic gene expression. And then you can amplify it or refine it based on this, what's called a reaction diffusion process. Okay. So people haven't actually found these morphogens in actual biology. We're still looking for them. But it could be something that could generate complex patterns. And one of the ideas then is to actually, why don't we actually use this to manage complexity on our chips, right? Because guess what? We get the noise for free. The transistors come out, they're crappy, and they're only getting crappier. Okay? And the fabrication guys, they don't charge you any money for that. Okay? And so this is something that we decided to try. Okay? And so the idea was here that we build this model of the cortex chip, of the cortex called the bump chip. And basically all the circuits on this chip are designed identical. Okay? We just replicate them. But when we send it to the fabricator and it comes back, well, it's like the hot sauce in your burrito. There's some places end up with a little bit more hot sauce than others, and you bite it and you're like, oh, this is really spicy. And either you make your transistors bigger, like you take bigger chunks of the burrito, so you average out the hot sauce, or you live with it. Okay, and so we decided to live with it. Some of these transistors are going to have lower thresholds, higher thresholds, make more current, less current. That's like the different amounts of dye and bleach, and we can then generate a pattern based on that, based on this Turing's process. And so this is just showing you an example of what that mismatch pattern looks like. If you plot the current levels in transistors that are identical, you get this sort of random pattern. When you're operating the transistors down in the subthreshold region where they're passing low currents, and they're exponentially sensitive to the voltage mismatch. And this is why people operate above threshold, where a square law is less sensitive, but then you burn more power, And it's the whole problem that we're talking about. And so here you go, you get all this mismatch. That's like your noise that's going to seed your pattern. And now we have to model this reaction diffusion process. And whereas Turing's model used these chemicals or morphogens, the idea here was just use electrical activity itself to represent those chemicals or those morphogens. And so we have two kinds of electrical activity: excitatory activity, which is like the dye that spreads a short distance, and inhibitory activity, which is like the bleach that spreads a long distance. And we put that in, and then we seed it with this mismatch, which we get for free from the fabricators. And voila, we get orientation maps. Okay, so this just giving, this chip has 10,000 neurons on it. And so here is a movie of the chip and what we call an orientation map. And I'll show you, I have to explain this a little bit. It's a little bit technical, but the idea is here is the actual activity that you see on the chip, which is supposed to look like these spots of a leopard or something like that. So you've got this guy here is making a lot of ink, and then the bleach is bleaching out all those guys, and you've got these spots and so on. That's just the excitatory and inhibitory interactions that we've put in the chip. And then now what we do is we do the same experiments that people do in cortex when they figured out these orientation maps. They present gradings of different orientations. And if we present this orientation and we get activity here, then we paint those cells in this map down here blue, right? So blue orientation preference is color-coded down here. Okay, and so you're just going to see the map being built in real time. As you're presenting these gradings, we are labeling the cells down here, and you see the map build up. Okay, so we present the yellow, everything gets yellow because there's no beta response. And then we present the different colors and so on and so forth. And we find that we find cells that are selective for actually different orientations, but we didn't actually hardwire this into the chip. Okay, we got it for free just from this activity-independent mechanism. And so I'm going to show you a picture here of a real orientation map that was taken. They actually just exposed the skull, the brain, and then they image directly activity of the brain, and they generate these maps the same way. Different orientations, you label the pixels different colors. And this is a single condition map. This is what it looks like when you just present it horizontal, for example. And dark regions are more activity, bright regions are less activity. This is really filtered because they are imaging thousands of neurons at a time. The resolution is not that great. And so you get the idea. We can generate these kinds of maps with the same features on this chip. Okay. So that's how far we've gone with this metamorphing or metaelectronics project, trying to actually translate these developmental rules into silicon so we don't have to wire up every transistor by hand. We can either get the chip to change its wiring, or we can seed these patterns spontaneously. Okay, and so the idea, I mean, you can read more about this in this article I wrote that came out last May, 2005, in Scientific American. And I would like to acknowledge the students who did the work, in particular the neurotrophin chips, cells, neurons that fire together, wire together, was built by Brian Taber, and that's Brian over there, and Paul Marola, who's over here, did the cortex chip with the orientation maps. And the retina chip was done by Kareem, who had graduated when we took this picture. So thank you. So we'll take some questions. I think we have maybe 5 minutes. Yeah?
Do you think you could use similar concepts to actually describe general purpose processors as opposed to silicon that mimics nervous system? In the sense that could use this nature efficiency to actually make it cheaper to design
Yeah.
a general purpose processor?
That's the $64 question, right? So the question is, can we actually, in a sense, what's the programming model for this, what I've described? Can we, in a sense, craft this on top of the existing programming model that we have? Is that fair? Or how do you? And my answer to that is that I think that we have to change the underlying hardware. Okay, I think that essentially we all know. I mean, I guess people who do hardware here know if you get you can do things in software, But if you have a lot of volume and you can hire a chip designer who can actually design an ASIC to actually solve your problem, that's going to be really orders of magnitude less power, 2 or 3 orders of magnitude less power. And it will allow you to put something in a cell phone that you've been doing on a computer or something by doing that. And there's also FPGAs people have been using to do this. And what the chip designer is doing is he's hardwiring that software into the chip. Right? These algorithms are ways of thinking about how to take things and hardwire them into the circuitry. Right? And so you want to be able to program the computer at the level of individual connections in order to get the most efficient implementation. Unfortunately, you don't have enough chip designers out there, and they are too expensive to do this. And so the idea is to think more about rules that can actually customize the wiring for you, which is what biology does. And so it's at a different level, right? You're actually rewiring the hardware. You're not leaving the hardware fixed and changing the instructions. The idea is to actually be able to customize the hardware directly. And eventually nanotechnology allows us to grow wires and things like that, and we'll be able to. So if we have these rules, we could actually be doing it for real. Yeah. Was there another? Okay. Okay, sure.
So there's also the question of reliability, right?
Yes.
When you see of the 800 engineers that you had for the Pentium core, I'm guessing probably 700. There are 5 lead architects, 700 verifications, and 95 actual circuit design.
Yeah.
So now they want to make sure that whatever they design is what they're
Yes.
supposed to do. But in this case, I guess it's going to try to make sure it's actually doing.
Yeah.
Whatever.
Yeah, that's something also that has to change. Does anybody have one of these optical mice? You guys use this? They've taken over, right? Yeah. Inside each of these optical mice is a little chip that detects motion. Okay. It's a little motion chip in each of these mice. Has anybody taken one of those chips apart? No, okay. It's really cool. It does 1,000 frames per second, and it's computing motion, and that's how you move your mouse. But if you take one of those chips, and they are based on these ideas, they've hardwired into the chip this motion algorithm based on actually how the fly does it, computes motion. If you take one of those chips and you look at the imager, it's a shitty image. There's about 30 by 30 pixels in there, and half of them usually don't work. And you're still selling you these mice. You're still buying them. Okay. And so it's a very different game if you're trying to do a computation autonomously to make a decision than it is if you're trying to make a computation or a pretty image for people to look at. Okay. And so these kinds of systems that we are building, right, if they are going to actually be recognizing people and things like that and reacting and doing stuff, right, the reliability question is whether it gets the answer right or not. It's not a question of characterizing every single element, right? You want to get the performance at the system level. Once the thing adapts or lands, it's doing the job right. That's what you're looking for, right? You're not looking for the traditional engineering ways that you make every element really precise and then you wire it up, and then by the time half of the pixels in here don't work. Okay, they still sell these mice. Okay, their yield on those chips is 100%. Okay, so that's different. Okay, and whether you like it or not, it's getting that way because now we already have error correction in all our ROMs, our USB keys, and things like that. But now it's getting to the point where the processors themselves need error correction, and so it has to be built in to the system, not added on. Which is how biology does it. The neurons are not so great, but they do a good job. Thank you.